Hey, good morning, everybody. Today we have we thank Carlos to be here. He's originally from Seville, and indeed he did his PhD here in the house in the EBD. Although then he moved to Sweden with a Ramon Areces fellowship, followed with Juan de la Cierva in the EP in the Pyrenees, and then in 2023 he came back in the EBD with a Ramon y Cajal fellowship, and he works in the interface between ecology, evolution, and behavior, especially I guess in birds. And that's all for me. I guess that many people will be online just because the weather. So also we will read questions from the YouTube emission. Okay, so thanks a lot, Carlos, and the floor is yours.
Okay, thank you. Thank you for the introduction and also for the opportunity to give this seminar. It feels literally at home for me. First, I have to apologize if you see me sweating and nervous, and it's just because I had a small car accident 20 minutes ago. While I was driving through the Cartuja, a street light fell down and crashed on the street. So I went through the remains, and both tires exploded. It was only 20 minutes ago, so I'm still a bit nervous about that. So my apologies. It's totally true. I'm not lying. Okay. So having said that, I would like to take this opportunity to introduce myself, especially to those of you who don't know me yet. So I'm gonna explain a bit about my background. I will try to provide an overview of the type of research I've done in the past. And I will conclude by describing my current and future plans at the EBD. So I have been linked to Doñana for the last 23 years, I think. And the reason is that I arrived there as a volunteer for the first time when I was 16. And I ended up working as a field technician and a bird ringer at the Spanish Ornithological Society, even before I obtained my undergraduate degree at the University of Seville in 2007. And after that, I wanted to do science, so I tried to get a PhD fellowship, but I failed. So I moved to Ecuador to manage a project for the Ecuadorian government about waterfall monitoring. I had some experience in aquatic birds that I had gained during my time in Doñana. So I joined BirdLife International to develop this project. But after that, I came back to Spain, and I again tried to get a fellowship, but I again failed. So I decided, together with some colleagues, to set up a long-term, well, by that time it was not that long, but actually, it is still running. So we set up a long-term longitudinal study on red-necked nigers in Doñana. After that, because I needed some training to do science, I obtained my master's degree in conservation biology at UPO. And I finally managed to get a fellowship. I got a Severo Ochoa fellowship to do my PhD. And I joined the EBD for 4 years. I did my PhD here. And then I got my first postdoc contract at the UPO again. I moved to Lund, as you said. Then I got the Juan de la Cierva fellowship, and I'm back to Seville as a Ramon y Cajal researcher. Over the course of my life, I have used different study models in addition to nigers, primarily birds, because I'm mainly interested in them. But I have also used bugs or fish, depending on the research question I wanted to answer. And as you can imagine from the high mobility of those organisms, I'm mainly interested in movement. I've done research in all types of movement, from foraging to migration and natal dispersal, breeding dispersal. But perhaps the topic I like the most is natal dispersal, which is the movement from the natal site to the site of first reproduction. And I like this movement because it has a very peculiar but simple feature, and is that because of movement of individuals, the location of reproduction is different for parents and offspring. And that has enormous ecological and evolutionary implications that I'm not going to explain right now, but I'm sure you all know. So I focused most of my research on natal dispersal, which is a very complex process consisting of 3 different stages: the initiation phase or departure, the transient phase or transport, and the termination or settlement stage. And I have mostly focused on this last one stage because it is where habitat choice takes place. And I'm very interested in the mechanisms driving habitat choice decisions of organisms as the final stage of dispersal. So I've been investigating the proximate means of the habitat choice mechanisms, the outcome, and especially also the contribution of each of mechanisms to population divergence. Briefly, the habitat choice mechanisms that we know now are 3, still recovering from the accident. The first one is direct genetic habitat choice, by which individuals decide where to settle based on habitat preference alleles. It is slow because fixing alleles takes time, so it is most important during the late stages of population divergence. We also have plastic habitat choice. This is a kind of imprinting by which individuals decide where to settle based on their previous experience. So it is plastic by definition, it is quick, and it is therefore important in early stages of divergence. And a specific, very specific form or mechanism of habitat choice is matching habitat choice, by which individuals decide where to settle based on a performance assessment of their own performance, provided their phenotype. So they assess different habitats, they assess their performance prospects, and they decide to settle in the habitat that maximize their fitness. So this is also a very quick process driving phenotype environment correlations, so creating population structure, and it is mainly important during early stages of divergence because of its rapidness. And why matching habitat choice is so peculiar? And it is because it challenges their general assumption that the dispersal counteracts the effects of divergent selection and therefore constrains local adaptation. And that's because by matching habitat choice, we do not assume that dispersers are a random subset of the source population. Dispersers are different from non-dispersers usually. And we now know that body size, body condition, body color, big shape, personality affects the dispersal propensity of individuals, and that results in a difference between dispersers and non-dispersers. And if individuals disperse according to their own phenotype to the environment that best match their phenotype and therefore increase performance, then dispersal should reinforce and not necessarily counteract local adaptation, and should eventually create population structure. So to evaluate all these mechanisms, I did a whole PhD using the study launched by Professor Jaime Poti, which was here at the EBD as well until he retired 4 years ago, I think. This is a long-term study on pied flycatchers located in central Spain. Every year we go there, we capture all individuals. They breed in nest boxes that were provided for them to breed. So we go there, we capture individuals, we determine life history traits such as breeding date, clutch size, hatching dates. We count their chicks, we identify them, and we do that in two different plots. The oak forest, which is the original population where pine flycatchers were present before the study started. And there is a newly created population separated by 1 kilometer from the other one in a pine plantation. So there were no pine flycatchers there before nest boxes were set up. And what I have been doing is to explore differences in different traits between the two populations. So both habitats are quite different, and they differ in biotic and abiotic features. And a very remarkable thing is that 1/3 of individuals exchange between habitats every year because of natal dispersal. But despite this high level of exchange, there are differences between the two subpopulations in different traits, like egg-laying dates, tarsus length, the color of individuals, and even neutral genetic markers, suggesting that there is some phenotypic and genotypic population structure in the face of dispersal or gene flow. So to try to investigate the role of the different habitat selection mechanisms on the observed divergence, what we did is to perform a massive cross-fostering experiment. We changed over 1,000 individuals from their original habitat to the same one or to another. So we cross-fostered individuals both within habitats as a control and between habitats. And then we assessed whether they returned to the habitat they had been taken or they returned back to the habitat they were from. And what we found is that more than 70% of individuals returned to breed to the habitat they had been taken to, suggesting that they imprint on the habitat and this was independent of their genetic origin. So that led us to conclude that habitat preferences in pine flycatchers are not genetically determined, but they are mainly determined by their prior experience in the area where they are born. But still, there is a fraction of individuals that were changing habitats and coming back to their original population. And when you look at the phenotype of these birds, they are not a random subset of the population. Those moving from the pine forest to the oak forest are particularly small, while those moving from the oak forest to the pine forest are particularly large. And those are manipulated individuals, but if you look at unmanipulated individuals, the pattern is exactly the same. Individuals moving, especially in the pine forest, those are individuals that were born in the pine forest. Those that leave the pine forest and disperse to the adjacent habitat are the largest individuals in the population, while those that stay in the pine forest are the smallest individuals in the population. So there is clear phenotype-dependent dispersal in the pine forest. And we wanted to figure out why. Obviously, matching habitat choice is a possibility. But if you want to test for matching habitat choice, you need to look at fitness, because the main assumption of matching habitat choice is that individuals assess their performance prospects and then decide where to settle. So what we did is, well, and of course, it should be operating at the same time as imprinting, because we learned that birds got imprinting on the habitat they had been taken to. So we measured the fitness of individuals to test whether matching habitat choice has a motive to evolve and operate in our population. And what we found is that size doesn't really matter in the oak forest, but a few millimeters can make a big difference in the pine forest. Those individuals in the middle of the body size range achieved the highest fitness, measured by recruitment success in this case, while those at the extremes of the body size distribution had the lowest fitness. And if you remember, those that moved from the pine to to the oakwood were those that were largest in the population, and they really gain fitness by moving to the adjacent habitat. But those that stay in the pine forest, they lose fitness by staying there. This suggests that at least for a fraction of the population, matching habitat choice is not operating, because by staying there, they are not gaining fitness. Their fitness is not increasing. Why is this? Because tarsus length, which is a measure of body size, mediates the competition ability of individuals, and large individuals outcompete smaller individuals, as in many other birds. So we concluded that competitive exclusion is the main mechanisms underlying size-dependent dispersal in pied flycatchers. So there was no apparent road for matching habitat choice. Then, where is matching habitat choice? Is it common or is rare? Because it is so advantageous that it should be everywhere. So in theory, it should be everywhere because it promotes local adaptation. It allows individuals to escape maladaptation, so it can maintain genetic variation. It increases the chances of population persistence. It has implications for ecological speciation, metapopulation dynamics, niche evolution, and ultimately, it may even lead to speciation. But by that time, we had strong evidence in support of matching habitat choice from simulations, studies, and lab organisms. But we have almost no evidence, supporting evidence, from natural settings. So I tried to find matching habitat choice in nature. Why is so rare? So, well, it's maybe actually rare because of constraints on expression. Maybe individuals are not mobile enough to explore different habitats, or they are not able to select their preferred habitats due to competition, or there might be constraints on our ability to detect or test for matching habitat choice. Perhaps other local adaptation mechanisms are operating at the same time and are confounding the effect of matching habitat choice. Or perhaps there are other habitat choice mechanisms operating at the same time and obscuring its effects. Or perhaps performance trade-offs are not strong enough in some situations to favor matching habitat choice, because as I said, individuals need to assess their performance to make their decisions. If there are not performance trade-offs, there's no reason for matching habitat choice to operate. So I joined Professor Andrew Hendry's lab for a while, and I dusted off an old data set he had collected during his PhD some 30 years ago. And he had collected data on habitat selection and the phenotype of sockeye salmon in Alaska. And this is a very cool study system because males and female bury a nest, which is called a reed, and the females stay there for a long time trying to protect the eggs. Some of the salmon are breeding in deep waters, but there are other salmon that use shallow waters for breeding in the streams. And some of the largest fish in shallow waters get stranded and birds prey on them. So there is a trade-off between water depth and the size of the fish mediated by predation risk. So it seemed to be a good study system for testing for matching habitat choice. They're individually marked. We know the cause of death of those individuals. They are monitored daily by visual inspection of these markings. And the trade-offs emerge from the selectivity of birds for larger fish because they provide a larger energetic reward. And they do that in places where they find it easier to hunt salmon, obviously in shallow waters. So for large fish, it should be difficult to live long enough to protect their eggs in shallow waters because they are going to be eaten by birds, while in deep waters, which are the preferred sites of large fish, small fish are going to be outcompeted by larger fish, and they cannot protect their eggs properly. So we checked for the spatial outcome of habitat choice, which is a phenotype by environment correlation, and we actually found it. We found that both males and females selected water depth in their breeding sites according to their body length. The larger you are, the deeper you breed. And that had important fitness implications. This is lifetime reproductive lifespan, which is the number of days a fish has been taking care of the eggs. So the longer you take care of the eggs, the higher the survival of the offspring. And this is water depth and body length. So as you can see, for males, there is almost no effect of water depth or body length. But for females, the effect is huge. If you are a large females breeding in a shallow site, then your probability of producing offspring. So the cost of making a mistake is not uniform across all segments of the population. It is quite uniform for males. It doesn't really matter. They can move around the nest, but females can't. But the cost of making a mistake is very, very large for a female. If you are not in the right place given your size, then you're gonna lose fitness. So matching habitat choice has the motive to operate only in a fraction of the population, only in large females, while the others do not need to make matching habitat decisions. And we thought that this may be a major constraint on the evolution of matching habitat choice, because when you look at the population as a whole, you don't get any evidence of matching habitat choice or phenotype-dependent dispersal. But when you look at the different segments of the population, then you get that evidence. So that might be a reason why matching habitat choice does not evolve as frequently as expected in nature. And that's why we entitled our paper, Matching Habitat Choice, It's Not for Everyone. So, if it is constrained and it is difficult to detect, does it really exist in real life scenarios? And does it facilitate local adaptation as expected? So to answer this question, I joined Professor Pim Edela's group at UPO, and we performed an experiment using grasshoppers as a model organism. This species is quite variable in body color, and they occupy a recently built area where they were going to build houses, but they stopped the construction. So there were only roads, dark asphalt roads, and pale tiles on the roadsides. So we had environmental heterogeneity. We had a black substrate and a white substrate, and we had white grasshoppers and dark grasshoppers. And the color of grasshoppers is important for their survivals because they increase their camouflage through background matching. Darker individuals tend to use darker substrates because they are harder to detect. And this obviously creates the nice performance trade-off we need to test for matching habitat choice and for matching habitat choice to operate. There is a reason why if you are dark, should aim for darker substrates, a very strong reason in this case. So what we did was to explore if we found the spatial outcome of matching habitat choice, and indeed we found it. There was a pretty nice correlation between body color and the probability of using a dark on a pale substrate. But this could be due to different reasons. For example, phenotypic plasticity could make individuals change in color to resemble the color of the substrate. Or perhaps there might be differential mortality depending on color, so mismatches individual would be removed by selection. So in another study, my colleagues performed an hormonal experiment and a capture-recapture experiment, and they estimated that changing color takes 7 weeks, which is too slow for explaining the pattern we observed. And also, if we wanted to explain this pattern by differential mortality rates should be around 60%, and they were only 4%. So neither plasticity nor natural selection could explain this nice phenotype environment correlation. And that leaves matching habitat choice as the main candidate. And what we did to test for matching habitat choice, provided that individuals are assumed to evaluate the local performance based on their own phenotype, was to change their phenotype. So we painted them in either black or white. So we noted their original location. We went to the field, we captured them, we painted them, and we realized them, and then we saw what happened. And what happened was that dark painted individuals moved to dark substrates, while pale painted individuals moved to pale substrates. And it was independent on their original location. So it was not genetically determined. It was not because of their prior experience on those substrates. It was just only because their phenotype changed and they responded by selecting a matching substrate. So yeah, we finally demonstrated that matching habitat choice does operate in nature and that it facilitates very quickly adaptation to novel environments. But this is all about space. What's next? I started to think about time, because individuals are able to use space as a resource, but they can also use time as a resource. So at some point, they could disperse in time as opposed to space. Is it possible? Indeed, it is. There are so many examples of dormancy of propagules in nature. For example, the seed bank of plants. There are examples also demonstrating that seed dispersal in time can affect gene flow and population structure. There are also examples from animals, the statoblast bank in bryozoans, or the famous diapause eggs of Daphnia. They are all examples of individuals that remain in a basal metabolic state or in a state of dormancy, waiting until the conditions are optimal for their own phenotype. But this has a major disadvantage and compared to space, and is that we cannot travel back in time. We can only move forward. So if you need to assess different environments, it is easier if you focus on space, because you can move back and forth and assess the environment that best matches your phenotype. But how can you do that when you cannot travel back in time? Is matching time choice possible by analogy to matching habitat choice? So given that it is not possible to travel back in time, yeah. Better? No? ¿Sí o no? ¿Cómo va? Okay. So, no? ¿Ahora? Sí, la farola. Tengo que tener cristales todavía. ¿Ya? ¿Ahora? ¿Más o menos? Yo creo que está diciendo que sí por cumplir. Bueno. So, yeah, I'm gonna travel back in time. So I was wondering if matching time choice can actually operate in nature. And given that you cannot travel back in time, perhaps individuals may experience periodically the same conditions, so they can choose, not now, but they can, if they are predictable, they can choose the best timing for their activities, for example. And a good example of this predictable periodicity in environmental conditions on Earth is the lunar cycle. The lunar period is quite short, it's around what month, as you know, and it exposes organisms to the same environmental conditions periodically, every time, every month. So there is an opportunity for them to select the best timing for reproduction, migration, molt, whatever. And just when I was starting thinking about this idea and drafting my first manuscript about this paper came out. And this paper focused on the differential use of moon phases by barn owls, depending on their morphs. There are red owls and white owls, and they use moon phases differently. Red owls are mainly active during new moon periods, while white owls are mainly active during full moon periods. Why is that? That is because it is advantageous for them. When they look at, the authors look at the amount of food they provided to their chicks, they found that red morphs failed to provide a lot of food during full moon conditions. That explains why you should focus on the period when you perform best in terms, for example, of chicks provisioning. And of course, if you receive less food, you are not going to develop correctly. So the fledgling success of red owls was also lower during moonlight. So if they flecked during moonlight periods, their fledgling success was lower. So there was a fitness consequence of using time differently depending on your phenotype. So it makes sense to carefully choose the best time for breeding for these guys. If you are red, you should never breed during full moon periods because your fitness is going to be lower. And this is what actually happens in barn owls. This is a nice example that matching time choice may occur in nature. And what about nigers? Because nigers are still more lunar-philic than owls. They depend on moonlight to forage. This is a plot showing the foraging efficiency of our nigers in Doñana, measured as the volume of the stomach. We measured their stomach, and we have data from sunset to almost the morning. And what is happening here is that if there is full moon, they can continue forage all night long, but if there is no moon, they can only forage after dusk, and they stop foraging. So they can only forage properly during part of the cycle, when there is full moon. So there is an opportunity for them to decide when to time their activities. And the result of this trade-off is that the individual slope of body mass changes, is quite variable. So it changes from mass loss to mass gain. Some individuals gain mass and others lose mass. And it depends on the time of the season. If you look at the ability of individuals to increase their body mass by accumulating fuel for migration. And you correlate that with this is a measure of the amount of moonlight they had while we were measuring them for body mass. You see that moonlight is playing a quite large fraction of variation in fuel deposition rate. So most individuals which are foraging under full moon conditions manage to put some weight. But if the amount of moonlight is low, then there is a lot of variability. And we don't really know why. We accept suggestions. But there should be a trait here that is affecting the ability of individuals to gain mass even in the absence of moonlight. But when you look at the development rate of nestlings, which is a trait directly linked to fitness, and the reason is that nigers breed on the ground, so they are quite vulnerable to predation. So the faster they develop, the lower the risk of predation and the higher the probability of survival. So the faster you grow, the higher the probability of survival. And the ability to gain mass of chicks with the food provisioned by their parents is also explained by the amount of moonlight they receive. Not surprisingly, they synchronize reproduction with the lunar cycle. But again, we find that there is a fraction of individuals that are quite well at provisioning food to their offspring when there is no moonlight. And we don't really, we don't know why. So that's the main topic we are working on right now. We are tagging individuals with multi-sensor loggers, and we are exploring physiological parameters. We are using an ecographer to estimate their consumption ability. We are tracking their body temperature dynamics for a full year. We are tracking their migration parameters. So we are trying to collect data to figure out what's going on here. So, is matching time choice a crazy idea? Well, maybe, but it seems to be possible. It happens in nature, and the consequences are quite similar to matching time choice because it should promote local adaptation. If you are not adapted to a particular place, you can change your temporal use of the time. Instead of, you don't need to move, perhaps you only need to change your rhythm. It should increase population persistence chances. And of course, it has implications for ecological specialization, niche evolution, temporal niche evolution, and it may even lead to speciation due to allochrony. If different morphs, for example, do not interbreed because they do not coincide in do not overlap in time. So that's what I wanted to tell you. I'm open to questions and suggestions, and this is an overview of what we are doing. I hope you like it. Thanks a lot.
Thanks, Carlos, for this incredible talk that you made, and also thanks for the figures that were really amazing. Also, questions. Any questions in the room? Okay.
Thanks, really nice talk. I had a more of kind of an opinion question, or I don't know. So do you know much about, at least in the systems that you're studying, do you know a lot about how they perceive themselves and their environment? Like, I mean, for example, the grasshoppers, I mean, do they see themselves, and can they, relate that with their environment somehow? Or do you know how they do it?
Yeah. The first thing is that you don't need to perceive yourself, but you may, based on your perception on what you experience. For example, if you get disturbed when you sit on a green chair all the time and you get disturbed and you get disturbed, maybe you don't know if you are green or not, but you are getting disturbed every time. And what we did is to perform an experiment to answer that question. It is still unpublished, but we disturbed grasshoppers on different substrates, accounting for their own coloration. And they are able to perceive some parts of their body, but when they decide where to settle, they base their decision on their perception of disturbance. So I shouldn't be here because I'm going to be killed. So I should better go there. I don't know if I'm white or not, but I should better go. And that's how it works in grasshoppers.
Maybe that through changes in temperature?
Yeah, that's also because temperature plays a role in this system, especially for darker phenotypes, because solar radiation is so high. So there has been also experiments along that line. And it's a component of the environment that they may use to perform or to make matching habitat decisions.
So they can perceive the temperature of the substrate and maybe relate that to their own temperature depending on their color, because the radiation will be different.
That's another cue they may use. Or they may be just, I don't know, not specifically in the grasshoppers, but how do you know that you are not locally adapted? I'm hungry. If you are able to evaluate your internal state, we do that all the time. You are hungry, probably, right now. So if you are hungry all the time, then something is not working properly. So you can use that as a cue to move to another place until you are satisfied.
So did they move randomly after disturbance, or did they choose a particular substrate?
They choose a particular substrate. So
it's both things. It's the disturbance and the perception of themselves that are playing there.
Yeah, but they apparently, according to our results, the cue they use the most is not their own coloration, but the disturbance they perceive when they are disturbed. So this is a long treatment where we noted their habitat use before disturbance, and then we randomly, for different colors, we disturb them or not, depending on their own color and on the color of the substrate. And what they do is to go where they are quiet, regardless of their own coloration. So it seems to be a stronger or more important cue for them than their actual phenotype.
Can I ask another question? Go ahead. Okay. So related to the nightjars and this variability you were seeing during the moonlight phase, you were seeing that during, so the cumulative effect was that, that one. So those guys up there that do the best, so their offspring.
So
no, those guys.
These guys.
Those guys.
So
sorry.
We only know that they grow faster.
Yeah. So you were saying that you will check for physiological parameters and all this stuff. What about, could it be possible that there is some kind of spatial variation in the use of space, or perhaps another variation in the trophic?
Maybe. Maybe
Diet,
we are also.
They are using that differ somehow, and that those guys can explore these different spatial trophic niches differently. Yeah,
they might be diet specialists. I guess they might be using different prey depending on moonlight conditions, because they are really constrained to detect prey when there is no moon. So if I were a niger, I will need to catch larger or lighter prey when there is no moonlight, because if there is no background light, then they would be better able to detect larger prey. So perhaps it has something to do with diet, but we need to explore that. Thanks.
Cool. Thanks.
Thank you for your talk. Very interesting. Carlos, I wonder if you find found an explanation how Ficedula that have matching habitat choice in different habitats, no? Pine forest or oak forest. Did you find an explanation for that? I'm thinking, did you prove that there was more food reserve in oak forest, and it's because the juveniles choose more stay there, or lost predation? I don't know. I wonder if you find an explanation of that.
Yeah, it's a complex story because indeed we did not find support for matching habitat choice. It seems that their decisions are based on their competitive ability, or, well, not the decision. The use they make of space is based on their competitive ability. If you are large enough to defend and acquire territory in the preferred habitat, then you go there and you acquire a territory. But it's, I said, it's a complex story because the preference of pine flycatchers for the oak forest changed throughout the study, and it's because breeding densities became similar and they are attracted by conspecifics. So the higher the number of conspecific is, the better the habitat is perceived by them. So, and because breeding density has been increasing, their perception of habitat quality has been changing accordingly. And what happens actually is that larger individuals that manage to get a place at the right or at the preferred site, they fail because they are so large that they are the subject of competition and fighting from others all the time. So their fitness is quite low. That means that dispersal occurs, but gene flow is not happening because they fail to produce offspring. So there is not an equivalence between dispersal and gene flow in this population. And we see very clearly the effects when you look at evolutionary dynamics, ecological effects, and that's more or less the way it works.
Okay.
Gracias. Thank you, Carlos, for your very, very inspiring talk. Yes, only a very general commentary is related to the use of your framework, initial framework of habitat choice, to the time choice, especially if we are thinking in the changes associated to global warming that are going to mismatch probably many systems, both that probably you know very well in birds and so, but also in other groups. I wanted to know your opinion of this field of research associated to the time matching.
Okay. Yeah. So that's that's one of the reasons I think that that investigating matching time choice is worthwhile, and it's because it may allows for local adaptation in a period where, because of climate change, the phenology or the timing of phenological events is changing a lot. So if you are able to quickly adapt to those changes, then you should probably increase your fitness, and then you can escape maladaptation. The main problem I see here is that you cannot travel back in time. So for seasonal events, then you should, or you may need to wait until next year. So this may apply to long-lived organisms only, for example, or to sedentary organisms. So that's something we need to solve, because a year is too long for matching time choice to operate, perhaps. But there are other hidden cycles which are acting within a year that may be helpful for them, like the lunar cycles and other biological rhythms that are hidden and we don't know, we don't pay attention at.
One?
regarding your first results, do you think that because the oak forest was the origin, the source, do you think that this historical background or like of the population might have also a role of selection acting only in the pines or and not found it in the oak forest?
Not really. And the reason is that we used this experiment to track individuals during their lifetime, and we measured their lifetime reproductive success. And because they move naturally between habitats, so we moved them, but they move naturally as well. So we had 4 groups, individuals that had local experience but not local genes, and then came back to the same plains they had been transferred or they originally came from. Those that had local experience but not local, well, you know, it's like a full factorial. And what we found is that experience is the main factor driving their lifetime reproductive success, the differences in lifetime reproductive success. So if they have the local knowledge about predators, food availability, or whatever, that I don't know, but if they developed locally during early life stages, they are better at exploiting that habitat. And that's what I know. So that argues, again, a role of an ancestral habitat or a genetic background of their fitness in one or another habitat.
Any more?
So, thank you very much.
Yeah, thank you very much. I think that's all right. Thank you again.